Okay, let's unpack this. Have you ever paused to wonder how a single microscopic cell, holding virtually the exact same genetic blueprint as every other cell in your body, knows precisely whether to become a beating heart cell, a complex neuron in your brain, or maybe a protective skin cell? Or, even more dynamically, how an entire organism can pivot, almost instantly, to respond to a sudden dramatic change in its environment, or mount a swift defense against a disease? Today, we're taking a truly deep dive into one of the most fundamental yet endlessly fascinating processes in all of biology, the intricate world of gene expression and its exquisite regulation. Our mission is to navigate this complex landscape, exploring the dance that turns genes on and off, or fine-tunes their volume, to build and maintain every living thing. By the end of this deep dive, our aim is for you to not just understand this information, but to truly master it, thinking about these molecular orchestrations like the elite professionals in the field.
What's truly fascinating here, and what we'll uncover, is how every organism, from the simplest single-celled bacterium to us, complex humans, achieves such precise control over its genetic information. It's profoundly more than just having the right genes encoded in our DNA. It's fundamentally about when, where, and with how much intensity those genes are expressed. This deep dive will illuminate the remarkable, tiny molecular mechanisms that govern these absolutely critical decisions, tracing the path from the initial genetic blueprint all the way to the final functional protein product.
We've got some incredible source material today to guide our journey, drawing extensively from Harper's Illustrated Biochemistry, specifically Chapter 38, which focuses on gene regulation. So get ready for some genuine aha moments as we break down what's happening at the molecular level, revealing surprising facts and connections you might not have considered before.
Yeah, and if we connect this to the bigger picture, a molecular understanding of these processes isn't just academic, it's paramount for human health. When gene expression goes awry, when it's dysregulated, it can directly lead to human disease. Think about cancer, developmental disorders, or even how pathogens like viruses hijack our cellular machinery. This means that a deep, comprehensive understanding of these regulatory mechanisms can directly lead to the development of powerful new therapeutics. These aren't just band-aid solutions. They're designed to alter the underlying biological problems, the pathophysiologic mechanisms, or even inhibit invading pathogens at their core.
Okay, let's start with the big picture then. Why is regulating gene expression so incredibly important? It feels like more than just a biological detail. It feels like, well, the very essence of how life adapts and thrives.
You've hit on a foundational principle there. Consider this. The genetic information, the complete DNA blueprint in nearly every normal somatic cell, that is, every non-reproductive cell of a complex multicellular organism like us, is practically identical. We're talking about the cells in your liver, your brain, your skin, all containing the same set of genes. The very few exceptions are highly specific, like certain immune cells that intentionally rearrange their genes, or in disease states like cancer, where chromosome integrity can be severely altered. But the vast majority of the incredible differences we observe in specialized cells and tissues, why a neuron acts like a neuron and a muscle cell like a muscle cell, or how an organism responds to the dynamic challenges of its environment, is almost entirely due to precise changes in when, where, and how much those genes are expressed. It's about modulating the amount as well as the spatial and temporal patterns of gene activity.
Right, the timing and location are everything.
Exactly. And from a biomedical perspective, this understanding is monumental. Organisms are constantly altering gene expression. Think about genetic developmental programs. That miraculous journey from a single fertilized egg to a fully formed organism with distinct organs and tissues is orchestrated by exquisitely timed gene expression. Or consider environmental challenges. A simple bacterium adjusts its metabolism based on nutrient availability, and our own bodies launch complex responses to stress or infection. And as we just touched upon, when this fine-tuned system goes wrong, it's intrinsically linked to disease. The mechanisms often begin with changes in gene transcription, that initial step from DNA to RNA. But as we'll explore, they are far, far broader than that. Correcting these misfired gene expression pathways is truly the frontier of medicine.
That makes perfect sense. And it's not just one simple on-off switch, is it? It sounds like a whole cascade, almost like a series of checkpoints where control can be exerted.
Precisely. While transcription control, that initial copying of DNA into RNA, is absolutely central, gene expression can be modulated at numerous levels. Each level offers a unique point of intervention and fine-tuning, giving the cell remarkable flexibility. Think of it less as a simple light switch and more like a multi-layered security system for controlling information flow.
Okay, so what are some of these layers?
For instance, one level is gene copy number. This is targeted modulation where a cell literally increases the number of copies of a particular gene. This happens when there's an incredibly high demand for a specific gene product.
Like making more blueprints when you need lots of something built quickly.
Exactly. Then there's gene rearrangement, a more dramatic approach where segments of DNA are physically moved or recombined. This can profoundly change how a gene is expressed, or even alter the protein it produces. After these genomic level changes, we get to transcription itself, the fundamental process of converting DNA into an RNA message. This is often seen as the primary decision point.
The main gatekeeper.
You could say that. But even once the RNA is made, the story isn't over. The next level is mRNA processing and transport. The freshly synthesized RNA needs to be chemically modified, things like adding a cap at one end and a poly-A tail at the other. Crucially, non-coding sections called introns are precisely spliced out to create the mature messenger RNA. And then this mature mRNA must be accurately transported from the nucleus, where the DNA resides, out into the cytoplasm, where proteins are made.
So processing the message and getting it delivered correctly.
Right. Each of these steps is a potential regulatory checkpoint. Beyond that, we have translational control, which regulates how much protein is actually synthesized from the mRNA. A cell can have plenty of mRNA, but still tightly control how much of it gets translated into protein. Then, once the protein itself is made, its journey isn't finished. Protein subcellular compartmentalization ensures the protein arrives at the correct location within the cell to perform its function.
Gotta get it to the right place.
Then there are post-translational modifications, PTMs, which are chemical changes, small tags, or alterations added to the protein after it's been synthesized. These PTMs can dramatically activate, inactivate, or alter a protein's function. And finally, the cell controls protein stabilization or degradation, dictating how long a protein remains functional before it's broken down. If a protein is needed only briefly, it's quickly degraded. If it's needed for the long haul, it's stabilized.
Wow.
All these positive and negative controls work in concert to increase or decrease the amount and/or activity of the final gene product. It's an incredibly intricate system, fine-tuned for maximum efficiency and adaptability.
That truly highlights how sophisticated the system is for enabling cells to adapt and differentiate. But at its most fundamental level, what's the core on or off mechanism at play? The simplest way to think about this regulation.
At its simplest, gene regulation is either positive or negative. Imagine a water faucet. Positive regulation is like turning that faucet on or increasing the water flow. Gene expression is increased because a specific regulatory element or molecule, which we call a positive regulator, enhancer, or activator, is present and doing its job. These are generally required for optimal gene expression.
Okay, the accelerator.
Right. Conversely, negative regulation is like turning the faucet off or diminishing the flow. Gene expression is diminished by a specific regulatory element or molecule, what we call a negative regulator, silencer, or oppressor. These molecules prevent or reduce gene expression.
The brake.
Exactly. But here's a neat trick, though. Sometimes a double negative can effectively act as a positive. If a specific molecule, an effector, inhibits the function of a negative regulator, it effectively removes the brake on gene expression, leading to an increase. So while it appears that gene expression is being induced at the molecular level, we call this de-repression the removal of repression.
Inhibiting the inhibitor.
Precisely. To simplify, if a regulator is present, negative regulation decreases the rate of expression, while positive regulation increases it. But if that regulator is absent, negative regulation increases the rate because the brake is off, while positive regulation decreases it because the accelerator isn't there. It's all about the precise dance of presence and absence of these specific molecules.
So the cell doesn't just have an instantaneous on-off switch. It can have wildly different temporal responses to a signal depending on the situation. This is where it gets really interesting, showing just how dynamic and context-aware these systems are.
Exactly. Biological systems exhibit three distinct types of temporal responses to a regulatory signal, and these patterns profoundly dictate how a cell reacts to a sustained or even a transient signal over time. It's not just if a gene is expressed, but how its expression profile changes over time.
What are these types?
The first is a type A response. This is characterized by an increased level of gene expression that is directly dependent on the continued presence of the inducing signal. Think of it like a push-button light switch. The light stays on only as long as your finger is holding the button down. When the inducing signal is removed, gene expression quickly diminishes back to its baseline level.
Okay, simple cause and effect.
Right. But importantly, the amount of expression reliably increases again every time that specific signal reappears. This type of response is incredibly common. In simpler organisms like prokaryotes, we see it in response to sudden changes in nutrient availability. In more complex organisms, it's observed after exposure to inducers like certain hormones, growth factors, or specific nutrients. A classic example we'll jump into very soon is the lac operon induction in bacteria, where they only bother to make enzymes for lactose metabolism when lactose is actually available. It's all about efficiency.
Makes sense. What's next?
Then we have the type B response. Here, you see an initial surge in gene expression, but it's transient, even if the regulatory signal persists. Imagine that smoke detector analogy again. It blares loudly for a minute when smoke appears, then goes silent even if the smoke is still there, but it resets and can blare again if the smoke briefly clears and returns.
So it adapts or desensitizes.
Exactly. This phenomenon is often called response desensitization recovery. It characterizes the action of many pharmacological agents where the body adapts to a drug over time, but it's also a feature of many naturally occurring processes. We commonly see this during the development of an organism, especially when only the temporary appearance of a specific gene product is required, even if the inducing signal might still be present. It's a smart way to ensure that cellular processes don't run indefinitely when only a temporary output is needed, preventing wasteful overproduction.
Clever. And the third type.
Finally, and perhaps most dramatically, there's the type C response. In this pattern, the regulatory signal acts purely as a trigger. It initiates an increased extent of gene expression that then persists indefinitely, even after the signal itself has completely vanished.
Like flipping a permanent switch.
Exactly. Once expression of the gene is initiated in the cell, it cannot be terminated, not even in subsequent daughter cells through many divisions. It's an irreversible and inherited alteration. Think of throwing a main circuit breaker in your house. Once it's flipped, the lights stay on even if you let go of the switch. This type of response typically occurs during the differentiation of cells into specialized tissues or organs, like when a stem cell makes the fundamental commitment to become, say, a muscle cell or a neuron.
A point of no return.
Precisely. Once that decision is made, it's maintained through all subsequent cell divisions. A fascinating example of this, which we'll dissect in detail, is the lambda-phage genetic switch, where a virus makes an irreversible decision to either integrate its DNA into a bacterial genome and lie dormant, or to replicate wildly and destroy the host cell.
Okay, let's transition then. To truly master gene regulation and understand this intricate symphony, we absolutely have to start with the pioneers. So much of our foundational understanding came from studying simpler organisms, particularly prokaryotes like E. coli. What did these seemingly simple model systems teach us, and why were they so crucial for building our knowledge before we tackled our own complex biology?
That's a perfect starting point. The meticulous analysis in prokaryotic organisms like E. coli and even lower eukaryotes such as Saccharomyces cerevisiae, baker's yeast, and Drosophila melanogaster, the fruit fly, truly established the fundamental principles of molecular biology that we still rely on today. Think of the central dogma, the flow of information from gene to mRNA to protein. These simpler systems were incredibly valuable because they're much easier to manipulate genetically and biochemically in the lab.
Easier to work with.
Definitely. While eukaryotic regulation is undeniably far more complex, many core concepts discovered in these foundational systems directly translate, providing an essential framework for understanding even the most intricate systems in human cells. Now, prokaryotic systems have some unique features that were absolutely critical to these initial groundbreaking discoveries. One key concept is the operon. In prokaryotes, genes involved in a common metabolic pathway, for example, all the genes needed to break down a specific sugar are often organized together in a linear cluster on the chromosome. They're regulated by a single shared promoter and a single regulatory region. The lac operon is the quintessential example we'll explore. It's important to note that operons are very common in bacteria, but are generally not found in eukaryotes.
Key difference.
A very key difference. Closely related to this is the cistron, which is simply defined as the smallest unit of genetic expression, essentially a segment of DNA that codes for a single polypeptide chain. And then you have polycistronic mRNA, which is a single mRNA molecule that remarkably encodes more than one separately translated protein. For instance, the lac operon mRNA contains the instructions for 3 different proteins, all translated from that one messenger.
One message, multiple proteins. Efficient.
Very efficient for bacteria. This polycistronic nature is also not common in eukaryotes, where typically one mRNA molecule codes for just one protein. We also classify genes based on their expression patterns. An inducible gene is one whose expression dramatically increases in response to a specific inducing signal or an activator molecule. These genes typically have relatively low basal transcription rates, meaning they're mostly off until they're explicitly needed.
Only on when required.
Right. Conversely, genes with high baseline activity are often subject to downregulation by repressors. In contrast, a constitutive gene is expressed at a reasonably constant rate and is not subject to extensive regulation. We often refer to these as housekeeping genes because they encode essential proteins needed for basic, fundamental cellular functions that are always required.
Always on, like
Yeah.
the lights in the house.
Exactly. If an inducible gene through a mutation somehow loses its regulation and becomes constantly expressed, we call that a constitutive mutation. These classifications help us understand the dynamic control that allows cells to adapt and conserve resources.
Let's zoom in then on the lac operon, a truly classic example that revolutionized our understanding of gene control. Jacob and Monod's 1961 operon model, based on E. coli's lactose metabolism, was utterly groundbreaking. So how does this ingenious system actually work to manage the cell's energy resources and ensure it's not wasteful?
The lac operon is, without a doubt, one of the best understood regulatory mechanisms in all of biology. It beautifully illustrates the precise coordinate expression of three enzymes that E. coli needs to metabolize lactose. These are galactosidase, LAC-C, the enzyme that hydrolyzes lactose into its simpler sugars, glucose and galactose; lactose permese, LAC-Y, a transport protein that acts like a gate, bringing lactose into the cell; and thiogalactoside transhedrolase, LAC-A, whose exact function is still debated but is consistently co-expressed with the other two.
Three enzymes, one pathway.
Exactly. And these three genes, LAC-C, LAC-C, and LAC-A, are physically clustered together on the bacterial chromosome, along with their key regulatory elements: the LAC promoter, the LAC operator, and an upstream gene called LAC-E, which encodes the LAC-I repressor protein. This entire arrangement allows for their perfectly coordinated expression. When transcribed, they form one large polycistronic mRNA, meaning one mRNA molecule carries the coding sequences for all three proteins, each with its own start and stop codons for separate translation.
Okay, so walk us through the mechanics. What happens when there's no lactose around?
Right, let's break down the mechanics, starting with the basal state, which is when there's no inducer present effectively, no lactose in the environment. The LAC-I gene, which is located just upstream, is constitutively expressed, meaning it's always making a small amount of its product, the LAC-I repressor protein. 4 identical subunits of this protein spontaneously assemble to form a tetrameric LAC-I repressor molecule. This LAC-I repressor has an incredibly high affinity for the LAC operator locus. Think of it as a molecular superglue.
It sticks really tight.
Extremely tight. The operator itself is a very specific 21 base pair region of DNA that has a fascinating symmetrical structure, an inverted palindrome which the repressor precisely recognizes. When LAC-I binds to this operator, it physically overlaps the promoter region, which is a crucial DNA sequence where RNA polymerase normally attaches to begin transcription. By sitting squarely on the operator, LAC-I effectively blocks RNA polymerase from binding efficiently to the promoter.
So it's a roadblock.
Exactly. The result: very low, almost undetectable basal transcription of LAC-C, LAC-C, and LAC-A. There are normally about 30 repressor tetramers floating around in the cell, and this concentration is sufficient to ensure over 95% occupancy of the single LAC operator element, maintaining that repressed off state. The cell essentially says no lactose, no need to waste energy making these enzymes.
Makes sense. So what happens when lactose does show up? How does the cell switch gears?
Okay, now let's consider induction, which happens when lactose is present, or when a synthetic inducer like IPTG, isopropyl thiogalactoside, which induces but isn't metabolized, is added. Even when permese levels are very low, tiny amounts of lactose or IPTG are able to sneak into the cell.
Right.
Once inside, this inducer molecule binds directly to the LAC-A repressor molecules, both those already bound to the operator and those free in the cytoplasm.
The inducer finds the repressor.
Yes, and this binding triggers a significant conformational change, a shapeshift in the LAC-A repressor. This structural alteration dramatically decreases LAC-E's affinity for the operator by a remarkable 10,000-fold.
Wow, 10,000 times weaker.
It's a huge change. With the repressor's grip weakened to almost nothing, RNA polymerase can now far more efficiently compete with LAC-RI, bind to the promoter, and initiate transcription. This turns on the LAC operon genes, and it's a classic example of that type A response we discussed earlier. Expression increases only when the inducer is present.
And it happens fast.
Impressively fast. Lac-specific mRNAs are fully induced within about 5 minutes, and Borg-lactosidase protein reaches maximal levels within 10 minutes, with up to 5,000 molecules per cell. That's a stunning 1,000-fold increase over the basal level. The cell is now fully equipped to efficiently break down lactose.
But there's another layer, right? What if glucose is also around?
Yes. Here's where E. coli's intelligence really shines, with another layer of control known as catabolite repression, which occurs in the presence of glucose. E. coli is smart. It prefers glucose as an energy source over lactose because metabolizing glucose is simply more efficient. So even if lactose is present, if glucose is also abundant, the cell prioritizes glucose and keeps the lac operon largely off.
How does it do that?
Glucose actually inhibits the enzyme adenyl cyclase, which is responsible for producing intracellular cyclic AMP, or CAMP. A crucial point here is that CAMP is absolutely essential for activating another key protein, the catabolite activator protein, CAP, also known as CAMP regulatory protein, CRP. The CAMP complex is a positive regulator. It binds to a specific DNA sequence called the CAMP response element, CRE, located just upstream of the lac promoter.
So CAMP levels signal glucose levels.
Precisely. Low glucose means high CAMP. When bound, CAMP-AMP directly contacts the RNA polymerase alpha subunit. These direct protein-protein interactions dramatically facilitate RNA polymerase binding to the promoter, increasing transcription more than 20-fold beyond what the lac removal alone would achieve. So for maximal lac operon activity, 2 conditions must be met. First, glucose levels must be low, leading to high CAMP and CANT activation. And second, lactose must be present, which prevents laci from binding to the operator.
Dual control, a brake, and an accelerator.
Exactly. This dual control negative regulation by lacini, positive regulation by CEP-CAMP ensures that the cell only invests energy in metabolizing lactose when glucose is scarce and lactose is available. It's a precise environmental adaptation that screams energy conservation.
So it's a brilliant system. If the cell is starving for carbon and lactose is available, it's ready to jump into action. But if glucose, its preferred fuel, is abundant, it says, thanks, but no thanks, I'll stick with what I know best for now. That's a truly powerful feedback system, making sure the cell is incredibly efficient. What happens, though, if something goes wrong in this system? What are the implications of mutations in such a finely tuned piece of molecular machinery?
Excellent question. The elegant simplicity of the lac operon also means that mutations in its components have predictable and highly illustrative effects, which were crucial for scientists to initially deduce its function. If there's a mutation in the laci gene where the laci repressor protein cannot bind to the operator DNA, then the operon will be constitutively expressed. It will always be on, producing the lactose metabolizing enzymes regardless of whether lactose is present or glucose is low. The repressor simply can't do its job of putting the brake on.
Always on.
Conversely, if there's a mutation in the laci gene where the laci protein cannot bind to the inducer, lactose or IPTG, then the operon will remain repressed even in the presence of the inducer. The inducer can't bind to the repressor to cause that crucial conformational change, so the repressor stays locked onto the operator, keeping the genes off permanently. The cell is unable to turn them on when needed.
Always off.
Exactly. Similarly, if there's a mutation in the lac operator locus itself, such that the repressor can no longer bind to that specific DNA sequence, then, just like the first laci mutation, the operon will be constitutively expressed. The brake on transcription is broken, and the genes are always on.
Also always on.
Right. These clear consequences of specific mutations have been instrumental in confirming our molecular understanding of the lac operon. In fact, this system is so well understood, it's now widely used in biotechnology. Researchers commonly use IPTG induction of plasmids bearing the lac operator promoter to efficiently express recombinant proteins in E. coli.
From basic science to practical tools.
It's a testament to the power of studying these foundational mechanisms, showing how fundamental discoveries can have immense practical applications.
Okay, from bacterial metabolism, let's pivot to viral strategy, which offers another incredible example of conditional gene regulation. The bacteriophage lambda provides a molecular drama, almost like a life or death decision for the virus within its host cell. This sounds like something straight out of a sci-fi movie.
It truly is a captivating molecular drama, and it's a quintessential example of a type C response, that unique pattern where a signal acts as a trigger, leading to an irreversible inherited alteration. Bacteriophage lambda is a temperate virus, meaning it can choose between two completely distinct pathways shortly after it infects an E. coli cell. It injects its double-stranded DNA genome, and then a critical molecular decision point is reached.
Two paths diverge.
Exactly. One path is the lysogenic pathway. In this choice, the viral DNA actually integrates itself into the host bacterial genome, becoming what's called a prophage. In this dormant state, the viral DNA remains mostly silent, essentially lying low, and is passively replicated along with the bacterial DNA every time the host cell divides. This pathway is typically favored under poor bacterial growth conditions, almost as if the virus is making a strategic move to wait out unfavorable times, ensuring its survival by not killing its host.
Lying low. Makes sense. And the other path?
The alternative is the lytic pathway. In this scenario, the viral DNA immediately begins to replicate aggressively, making about 100 copies of itself. It then synthesizes viral proteins, assembles new virus particles, and eventually causes the lysis or bursting and killing of the host cell, releasing the new viruses to infect other unsuspecting cells. This pathway is typically favored under good bacterial growth conditions, where resources are abundant for rapid viral replication. The molecular switch between these two fates is a powerful and elegant example of genetic control at its finest.
What's the switch itself then, and what throws it, moving the virus from that dormant lysogenic state towards a destructive lytic cycle?
The lytic-lysogenic switch is governed with extraordinary precision and centers around an 80 base pair region in the lambda genome called the right operator, OR. This OR region is flanked by two crucial regulatory genes. On its left is CI, which encodes the lambda repressor protein, and on its right is CRO, which encodes the CRO protein.
CI and CRO, the key players.
They are the stars of the show. The key here is that when lambda is in its prophage, dormant state, the CI repressor gene is virtually the only lambda gene expressed. But if the bacteriophage enters polytic growth, the CI repressor gene is turned off, and the CRO gene, along with many other lipolytic genes, is expressed, so they're in a reciprocal relationship. When CI is on, CRO is off, and vice versa. These two genes are locked in a molecular battle for control, regulating each other's expression and ultimately determining the viral fate.
A molecular seesaw.
A perfect analogy. The 80 base pair or OR region itself isn't a single binding site. It's intricately subdivided into three discrete tandem 17 base pair DNA elements, OR1, OR2, and OR3. While their nucleotide sequences are similar, they are critically not identical, which allows for differential binding. Both the CI and CRO proteins can bind to these three sites, but they have distinct opposing affinities for each. The CI repressor binds most preferentially to OR1, then OR2, and least tightly to OR3, so OR1, OR2, OR3. It's a two-domain molecule with an N-terminal DNA binding domain and a C-terminal dimerization domain, and it functions as a dimer, binding much more tightly to the operator as such. The CRO protein, on the other hand, is a smaller single-domain protein that also binds as a dimer, but its preference is the opposite. OR3, OR2, OR1.
Opposite preferences for the same sites. Clever.
It is. What's more, this entire region also contains two promoter sequences that direct RNA polymerase to transcribe in different directions. One directs transcription rightward for CRO and other lytic genes, and the other directs transcription leftward for the CI repressor gene. This elegant molecular arrangement sets up the perfect scenario for a highly sensitive genetic switch.
So how does the virus actually decide whether to go dormant or start replicating? What's the initial trigger for each pathway, and how is that decision maintained?
The decision hinges entirely on the competitive binding and precise regulatory actions of the CI and CRO proteins, each acting on these very specific operator sites and promoters. Let's first consider the lysogenic state, where the virus remains dormant as a prophage. In this state, the CI repressor gene is the only lambda gene expressed. What happens is that CI repressor dimers preferentially bind to OR1 due to their highest affinity for that site. This initial binding then cooperatively enhances, by a factor of 10, the binding of another CI dimer to OR2. The affinity for OR3 is the lowest, so it's usually unoccupied in this state.
So CI helps itself bind better.
Yes, cooperative binding. This binding of CI to OR1 and OR2 has two major critical effects. First, there is negative regulation of CRO. The occupancy of OR1 by the CI repressor physically blocks RNA polymerase from binding to the rightward promoter, which effectively prevents CRO expression and, by extension, the expression of all other lytic genes.
Stops the lytic cycle genes.
Exactly. Second, and this is where it gets truly ingenious, there's positive regulation of CI itself. The CI repressor bound to OR2 actually enhances RNA polymerase binding to the leftward promoter, which conveniently overlaps OR3. This boosts transcription and expression of its own CI gene, This enhancement is mediated through direct protein interactions between the promoter-bound RNA polymerase and the OR2-bound repressor, much like the CKP interaction we saw in the lac operon.
Wow, so it turns off its rival and turns itself on.
Precisely. The lambda CI protein is a masterful dual regulator. It acts as a negative regulator by turning off CRO, and simultaneously as a positive regulator by enhancing its own expression. This creates a beautifully stable, self-perpetuating lysogenic state. The repressor ensures its own continuous production while diligently shutting down the lytic genes. And there's an additional clever feedback loop. If CI concentration gets too high, excess CI can bind to OR3, its lowest affinity site. This binding then diminishes transcription of its own gene by blocking RNA polymerase. This prevents overexpression and maintains a precise homeostatic balance of CI.
A repressor that activates its own expression. That's an incredibly clever feedback loop for stability, almost like a self-regulating thermostat for maintaining dormancy. But how then does it switch to the alytic cycle? What breaks this stable, self-sustaining state and throws the other switch?
The switch to the lytic cycle is triggered by environmental stress, specifically DNA-damaging agents like ultraviolet light. This is the crucial external signal that destabilizes the entire system.
UV light, like sunlight damage.
Exactly. When the bacterial host is exposed to such damage, it generates single-stranded DNA fragments. These fragments act as a critical internal signal to activate a bacterial coprotease called RHA. Activated RHA is the key player here. It's a remarkable enzyme that, when active, specifically cleaves the CI repressor protein. It cuts the repressor precisely between its two functional domains, the amino-terminal DNA binding domain and the carboxyl terminal dimerization domain.
Cuts the repressor in half, basically.
Effectively destroying its structure and function. This cleavage event is catastrophic for CI's function. It causes the CI dimers to dissociate. Since dimers bind much more tightly than monomers, this immediate dissociation of CI from OR2 and eventually from OR1, which has the highest affinity but relies on cooperative binding, occurs rapidly. The consequences of removing CI from these critical operator sites are immediate and profound. First, RNA polymerase suddenly gains immediate unimpeded access to the rightward promoter, and transcription of the CRO gene begins. Second, and just as critically, the enhancing effect of CI at OR2 on leftward transcription, which promotes CI expression, is lost. So not only are the lytic genes turned on, but the CI gene itself is simultaneously turned off.
The balance tips completely.
It does. Now we enter the lytic state, specifically the early lytic growth phase. With CRO transcription now active, newly synthesized CRO protein starts to accumulate. Remember, CRO's binding preference for the operator sites is the opposite of CI's, OR3, OR2, OR1, and crucially, unlike CI, there is no cooperative effect with CRO binding. The immediate consequence of CRO binding to OR3 is that it directly turns off transcription from the leftward CI promoter. This utterly prevents any further CI repressor synthesis, irrevocably shutting down the lysogenic pathway.
CRO shuts down CI for good.
Exactly. The switch is now completely and irreversibly thrown in the lysogenic direction. CRO is on and CI is off. As CRO concentration increases further in the cell, it will eventually occupy OR1 as well. This actually then reduces its own gene expression, which is a necessary fine-tuning step to properly regulate the later stages of the lytic cycle, ensuring the virus efficiently completes its replication and host cell lysis.
So an incredibly precise molecular trigger DNA damage leads to this irreversible inherited decision, perfectly illustrating that type C response. It's truly a masterclass in how conditional protein-DNA interactions and protein-protein interactions control transcription, leading to such dramatically different life pathways for the virus.
Indeed. It's a textbook example of how a molecular switch operates at such a fundamental level. Both CRO and CI, the key players in this decision, utilize a specific common structural motif for DNA binding called the helix-turn-helix HTH DNA binding motif, which we'll discuss in more detail shortly. This genetic switch of bacteriophage lambda alongside the lac operon provides an unparalleled understanding of transcriptional activation and repression at the molecular level. It powerfully demonstrates that regulatory proteins often have multiple distinct functional domains, separate parts of the protein responsible for DNA binding, for forming dimers, and for interacting with other crucial factors like RNA polymerase.
Modularity again.
Yes, this concept of modularity is a recurring and incredibly important theme throughout gene regulation.
Okay, so if prokaryotes are the brilliant soloists of gene regulation, our own eukaryotic cells, with their vastly more complex structures, are the entire symphony orchestra. The same basic principles of control apply, but with thousands of times more DNA, the complexity absolutely explodes, largely because of how that DNA is packaged and accessed. What are the big differences here that add all these new layers of intricacy?
That's a perfect way to put it. It's a critical leap in complexity. The two major differences in eukaryotes are indeed the sheer amount of DNA and perhaps even more importantly, the elaborate way it's packaged. Human cells, for instance, have about 1,000 times more genetic information than a bacterium like E. coli. Much of this extra DNA doesn't directly code for proteins. Instead, it's likely involved in the incredibly intricate regulation required for tissue differentiation, for the development of complex multicellular organisms, and for their sophisticated adaptation to highly diverse and challenging environments.
More instructions needed for a more complex machine.
Essentially, yes. It's the instruction manual for the conductor, not just the sheet music for the instruments. But the truly crucial difference is the packaging. Eukaryotic DNA isn't just a loose strand. It's extensively folded and compacted into a highly organized protein-DNA complex called chromatin. Histones are the key protein components of this complex. They form the fundamental repeating units of chromatin known as nucleosomes, where the DNA is precisely wrapped around a core of these histone proteins. This elaborate packaging isn't just for neat storage. It actively dynamically influences gene regulatory mechanisms by physically restricting access to the underlying DNA. It's a powerful barrier that must be overcome for genes to be expressed.
So chromatin isn't just a passive packing material. It's an active, dynamic player in gene control, like a molecular gatekeeper. How does it act as a barrier? And what are the clever ways cells overcome that to actually access the specific genes they need at the right time?
That's precisely it. Chromatin structure provides an additional, absolutely fundamental level of gene transcription control. Large regions of chromatin are often transcriptionally inactive, presenting a physical barrier that prevents transcription factors and RNA polymerase from easily accessing the underlying DNA. The incredible diversity of specialized cells and tissues in a multicellular organism, each with nearly identical DNA, is achieved partly by having different regions of chromatin open or closed for transcription in different cell types.
Different sections are accessible in different cells.
Exactly. For example, the aglobin gene cluster, which is essential for making red blood cells, is found in active open chromatin in reticulocytes, precursors to red blood cells, but is completely inactive and tightly packed in muscle cells. This means the DNA is packaged in a way that makes it literally inaccessible in cell types where that gene isn't needed. The constant formation and disruption of nucleosome structure is central to eukaryotic gene regulation.
How do cells control whether it's open or closed?
One of the most important ways cells manage this dynamic barrier is through histone covalent modification, which is often referred to as the histone code. Histones, those core proteins around which DNA is wrapped, are subject to a wide range of specific post-translational modifications, PTMs. These chemical tags are typically added to the amino terminal tails of the histones, which protrude from the nucleosome. These modifications are incredibly dynamic and reversible, constantly being added and removed, forming a complex language.
The histone code.
Yeah.
Like a chemical language on the protein.
Exactly. The best understood PTMs are acetylation and deacetylation, which occur on specific lysine residues. Acetylation, catalyzed by enzymes called histone acetyltransferases, HATs, adds an acetyl group to these lysine residues. This addition reduces the positive charge of these lysines. Why is that important? Because it weakens the histone's affinity for the negatively charged DNA, effectively loosening the DNA from the nucleosome. This loosening generally correlates with active transcription, or at least the potential for transcription, making the DNA more accessible.
Acetylation opens things up.
Generally, yes. But it's more than just charge. Acetylation also creates new binding or docking sites for other crucial proteins, particularly ATP-dependent chromatin remodeling complexes. These are multi-protein machines that contain structural domains specifically designed to bind to these modified histones. Once recruited, they use the energy from ATP to physically reposition, remove, or even alter the nucleosomes, which in turn dramatically increases the accessibility of underlying DNA sequences, like enhancer or promoter regions.
So they physically move the roadblocks.
They can. So co-regulator is a term that includes both chromatin modifiers like HATs and chromatin remodelers work in conjunction to open up gene promoters, making it easier for transcription activator proteins, RNA polymerase II, and general transcription factors, GTFs, to bind and do their job. Conversely, deacetylation, catalyzed by histone deacetylases, HDCs, which are often recruited by transcriptional core pressors, has the exact opposite effect. It compacts the chromatin and actively represses transcription.
Deacetylation closes things down.
Right. This system is so complex that we envision it in terms of code writers, the enzymes that catalyze these PTMs. Code readers, the proteins that recognize and bind to these specific PTMs, interpreting the code, and code erasers, the enzymes that remove the PTMs. This is very analogous to signal transduction pathways, with kinases, phosphatases, and binding proteins constantly communicating. Histone PTMs represent a highly dynamic and information-rich source of regulatory information, far beyond simple on-off switches.
Wow, so many ways to modify histones. It's like a secret language layered on top of the genetic code, constantly being written, read, and erased. This directly influences how DNA is presented to the transcription machinery. And the fact that some of these PTMs are linked to disease is incredibly powerful. Can you give us a few examples of these novel histone PTMs that really highlight their profound impact and connection to the cell's environment?
Absolutely. The field of histone PTMs is rapidly expanding, revealing incredible layers of information density, showing just how responsive our genome is. Beyond acetylation, we're discovering a whole lexicon of modifications, many of which provide direct links between cellular metabolism, environmental cues, and gene expression.
Like what?
For instance, lactylation is a modification where a lactate molecule, a byproduct of metabolism, is chemically attached to histones. This modification specifically promotes permissive transcription, meaning it helps turn genes on. What's truly fascinating is its relevance. It plays a crucial role in how macrophages, key immune cells, respond to hypoxia-low oxygen conditions. This directly links how active your muscles might be, producing lactate, to how your immune system responds to stress.
Metabolism directly affecting gene expression through histones.
Exactly. Then there are even more surprising ones, like serotonylation and dopaminylation. These are modifications where neurotransmitters like serotonin and dopamine, typically associated with brain chemistry, can chemically attach to histones. Serotonylation, for example, is permissive for transcription and has implications for neuronal differentiation, literally influencing how brain cells develop. Dopaminylation, on the other hand, is linked to altered transcription in contexts like drug-seeking behaviors.
Brain chemistry physically changing chromatin. Incredible.
Imagine the implications for neuroscience and psychiatric disorders, where brain chemistry directly influences which genes are active. And it doesn't stop there. Some modifications are non-enzymatic, meaning they happen spontaneously due to metabolic byproducts or environmental exposures. Take glycation, for example. This is a non-enzymatic reaction involving sugars, which can alter nucleosome stability. It's highly relevant in conditions like breast cancer and hyperglycemia, highlighting how systemic metabolic imbalances can directly impact chromatin structure and gene expression. We even see modifications like 4-opsonynylation and acrylane adducts, which are chemical additions resulting from lipid peroxidation products, basically, damage to fats in our cells. These are also non-enzymatic, lead to nucleosome destabilization, and are relevant in contexts like cigarette smoke exposure, showing how environmental toxins can directly impact your gene expression by modifying histones.
So our environment and metabolism leave marks on our chromatin.
It's like a living, breathing metabolic barometer for the cell. This incredibly diverse array of modifications underscores how sensitive chromatin is to the cell's metabolic state and external environment, and how these chemical tags directly influence which genes are accessible and active. Beyond histones, DNA methylation also plays a crucial role in chromatin level control. This involves the addition of a methyl group to specific deoxycytidine residues, typically at 5-arbo-MECPG3 helical sequences. This modification most often leads to a decrease in gene transcription. It essentially acts as a repressive mark. For example, in mouse liver, only the unmethylated ribosomal RNA encoding genes are expressed.
Methylation usually means off.
Usually, but not always. There's evidence that many animal viruses are not transcribed when their DNA is heavily methylated, possibly as a host defense mechanism. Conversely, the acute demethylation of these 5-MEC residues in specific regions of steroid hormone-inducible genes has been associated with an increased rate of transcription. However, it's important to note that, like with histone PTMs, it's not a universal rule that methylated DNA is always transcriptionally inactive, or that all inactive chromatin is methylated. The picture is far more nuanced, with methylation often working in concert with histone modifications.
It's a complex interplay.
Definitely. Finally, the physical act of transcription factor binding can also lead to direct nuclear sum disruption. When specific transcription factors bind to their particular DNA elements, they can literally push nucleosomes aside or prevent them from reforming, directly opening up the DNA. They can also recruit those multi-protein co-regulator complexes we just discussed, which then covalently modify and/or remodel nucleosomes. Ultimately, all these mechanisms lead to dynamic chromatin-level structural changes that either increase or decrease the accessibility of the underlying DNA to other factors, and crucially, to the transcription machinery. It's a constant dynamic interplay between protein binding and DNA packaging, a molecular tug-of-war for access.
Okay, so beyond the fundamental chromatin state, specific DNA elements act like sophisticated remote controls, boosting or dialing down gene expression, even from a great distance. Let's talk about enhancers. These sound like the ultimate master volume knobs for our genes.
Enhancers are truly fascinating and incredibly powerful regulatory elements. They are specific DNA sequences that significantly facilitate or enhance transcription initiation at a promoter, but with remarkable flexibility. Unlike promoters, which are typically located very close to the transcription start site, enhancers can exert their influence from tens of thousands, sometimes even hundreds of thousands, of base pairs away from a promoter.
Huge distances, how?
It's thought to involve DNA looping, bringing the enhancer physically close to the promoter in 3D space. What's even more surprising is that they work when oriented in either direction forward or reverse, and they can be located upstream to the 5-foot side, downstream to the 3-foot side, or even embedded directly within the transcription unit of a gene.
Incredible flexibility.
What's more, enhancers can be quite promiscuous. They have the ability to stimulate transcription of any promoter in their general vicinity, and a single enhancer might even act on more than one promoter, making their regulation quite complex. Crucially, enhancer elements themselves do not code for any product. They are active only when physically linked to the promoter on the same DNA molecule, what we call in cis.
They need to be on the same piece of string.
Right. Their profound effect is mediated by enhancer-binding proteins, which are special transfactors, meaning they're proteins encoded by genes located elsewhere in the genome that bind to the enhancer sequence. These enhancer-binding proteins then act as molecular bridges, interacting with a plethora of other transcription proteins, including coactivators, mediator complexes, and various components of the basal RNA polymerase II transcription machinery. Ultimately, the binding of these transfactors to the enhancer leads to increased binding and/or activity of the basal transcription machinery on the linked promoter.
They recruit the machinery.
Essentially, yes. Enhancers also often make the regions they reside in more sensitive to nucleuses, which is a strong indicator of open active chromatin. A recent discovery highlights the sheer scale and importance of some of these elements. Super enhancers. These are massive tandem clusters of various enhancer elements found ubiquitously in mammalian genomes. They're strongly associated with highly expressed genes that are absolutely critical for defining cellular identity, for example. What makes a liver cell a liver cell?
Super enhancers define who you are, cell-wise.
In many ways, yes, and they are thought to contribute significantly to the formation of biomolecular condensates dynamic cellular compartments that literally concentrate transcription factors and machinery, almost like transient molecular factories. So enhancers are not just remote controls, they can be superpowered, context-dependent remote controls that shape cell fate.
This concept of a remote-controlled DNA element is wild, and the fact that they can work in any direction over huge distances is just incredible. It makes me wonder, though, how does the cell achieve this without accidentally turning on every gene nearby? How does it maintain order and precision? And do you have a specific example that really brings this architectural feat to life, showing us how this remote control actually works?
That's a brilliant question, and it brings us to a beautiful and incredibly well-studied example of precise enhancer function. The up interferon enhancism. The interferon gene is a critical part of our cell's immediate antiviral response. It's rapidly and massively induced upon viral infection. Its enhancer is located relatively close to the gene, specifically between nucleotides -110 and -45 relative to the transcription start site, +1. This enhancer isn't a single element. It's a precisely orchestrated complex composed of 4 distinct clustered cis elements, each bound by unique transfactors.
4 parts working together.
Exactly. These 4 key factors are NFK, a well-known transcription factor involved in immune responses, a member of the interferon regulatory factor, IRF family, another crucial immune player, the heterodimeric leucine zipper factor, ATF2C Jun, and finally, a ubiquitous and abundant architectural transcription factor called HMG-IY. HMG-IY is absolutely critical here. It binds to specific A plus T-rich sites, 4 of which are interspersed throughout the enhancer, and it induces significant bends in the DNA. Think of it literally twisting and folding the DNA.
It bends the DNA.
Yes, significantly. The magic happens during the enhancosome formation. The precise DNA bending induced by HMG-IY is not just a random event, it's absolutely essential. It physically facilitates the cooperative formation of a unique 3D structure with the other three factors. It's believed that these precise DNA bends bring the other three transcription factors into close proximity, allowing them to make critical direct protein interactions that wouldn't be possible if the DNA were straight. This results in the formation of the interferon enhancosome.
The bent DNA forms a landing platform.
A perfect analogy. This meticulously organized, three-dimensional structure provides the exact appropriate surfaces and spatial organization for the efficient recruitment of key coactivators, like SwissNF, an ATP-dependent chromatin remodeler that can literally push nucleosomes around, and PCF, a histone acetylhortol-transferase that adds those activating chemical tags we talked about. It also efficiently recruits the general transcription machinery, RNA polymerase II and GTS. It's not just about which proteins are present, but their exact arrangement and interactions in 3D space, like a molecular puzzle where every piece fits perfectly. This meticulous assembly leads to a massive increase, over 100-fold, in belly interferon gene transcription upon viral infection, allowing the cell to mount a rapid and incredibly powerful antiviral response.
Wow, so the DNA literally bends to bring these pieces together, forming a perfect docking station. That's incredible. It's like molecular origami creating the ideal landing pad for all the necessary machinery. That's a true aha moment right there, showing how architecture plays such a direct and active role in controlling our genes.
Yes, it profoundly demonstrates that appropriate enhancosome architecture is strictly required for efficient transactivation. And similar enhancosomes, involving distinct sets of cis and trans factors and co-regulators, are thought to form on many other mammalian genes, indicating this is a general principle of eukaryotic gene regulation. Precise 3D organization is key.
What about the opposite, silencers?
Right. While enhancers boost gene expression, we also have their counterparts, silencers. These are cis-acting DNA elements that, as their name suggests, decrease gene expression. Less is known about their precise mechanisms compared to enhancers, but it's clear that, like gene activation, they also likely involve chromatin-level covalent modifications of histones and other proteins, mediated by silencer-recruited repressors and their associated copressors, which often bring in HDACs to compact chromatin.
Turning the volume down.
Exactly. The intricate interplay of enhancers and silencers also plays a major role in tissue-specific expression. Many genes harbor enhancers that can dictate their expression in a very specific cell type or tissue. This means the same gene might have an enhancer that activates it only in the liver and another that represses it in the brain. Researchers can rigorously test this tissue-specific modulation using a powerful technique called the transgenic animal approach. By fusing suspected tissue-specific enhancers or silencers to easily detectable reporter genes, genes that produce a visible or measurable product, and then introducing these custom-built DNA pieces into single-cell embryos to create transgenic animals, they can observe where and when the reporter gene is expressed, thus functionally mapping the precise tissue specificity of that regulatory element.
Testing it in a whole organism.
It's a very powerful approach, especially in developmental biology.
That's a clever experimental approach. But in the vast expanse of DNA, how do scientists actually pinpoint these elusive regulatory elements? It seems like trying to find a tiny needle in an enormous haystack.
It definitely sounds like it, but they use a powerful and widely employed molecular tool called reporter genes. The concept is quite elegant. A DNA fragment that is suspected of harboring regulatory sequences, such as an enhancer, a promoter, or any other response element, is ligated upstream of a suitable reporter gene. Examples of common reporter genes include firefly luciferase, which produces light like a firefly, human growth hormone, or chloramphenicol acetyltransferase, which modifies an antibiotic. These reporter genes are chosen because they encode proteins that are easily assayed, measured in the lab, and are not normally produced by the host cells, so any activity detected is directly attributable to the introduced experimental construct.
So you attach the suspect DNA to something you can easily measure.
Precisely. This custom-built DNA piece, the chimeric construct, is then introduced into host cells, typically in cell culture, via a process called transfection. If the inserted DNA fragment contains an enhancer or a specific response element, for example, a hormone response element, HRE, then expression of the reporter gene will increase under the appropriate conditions, for example, when you add the specific hormone to the cell culture. By systematically using progressively shorter pieces of the suspected regulatory DNA, introducing internal deletions, or creating specific point mutations within the sequence, researchers can precisely pinpoint the exact location and even the critical specific nucleotides within the response element that are essential for its function. It's a highly effective way to functionally map these regulatory regions.
Narrowing it down piece by piece.
Exactly. However, a caveat to this approach is that transfected plasmid DNAs may not always form the exact classical chromatin structures found in the native genome, which can sometimes slightly affect the interpretation of results compared to the true in vivo state. Nevertheless, this strategy has been incredibly successful, leading to the identification of hundreds of enhancers, silencers, tissue-specific elements, and elements responsive to hormones, heavy metals, and drugs, painting a detailed map of gene control. When we think about how combinations of elements provide diversity, it becomes incredibly clear that while prokaryotic genes often have relatively simple on-off responses to environmental cues, eukaryotic genes, especially in mammals, are far more complex. A single eukaryotic gene can be regulated by multiple complex environmental stimuli converging on it. The resulting responses can be highly variable. They can extend over a considerable range, exhibiting additive or synergistic positive responses that are often counterbalanced by negative or repressing effects.
More like a dimmer switch, or multiple switches than just on-off.
Exactly. Imagine a gene that is fine-tuned by a symphony of signals rather than just one. Furthermore, a single effector, such as a hormone, can activate some genes in a cell, repress others, and leave still others unaffected, all within the exact same cell type. This kind of flexibility requires a much more intricate regulatory arrangement than a simple on-off switch. The specific collection and precise organization of DNA elements within a promoter region, along with the associated transcription factors, dictate how a given gene will respond, to what extent, and how long that particular response will be maintained. It's like a highly customized multi-slider control panel for each and every gene.
With so many powerful enhancers working over long distances, sometimes tens or hundreds of thousands of base pairs away, how does the cell prevent them from accidentally turning on every gene nearby? How does it maintain such precise order and prevent this powerful remote control from becoming chaotic?
This is where the higher order spatial organization of chromatin becomes absolutely critical, and it's a rapidly evolving area of research. Chromatin isn't just randomly packed, it's intricately arranged into functional units or domains, often interacting with the nuclear matrix or other physical compartments within the nucleus. The International 4D Nucleome Project, for instance, is actively studying this macro-scale organization, aiming to understand how genome structure and dynamics relate to transcriptional activity in 3 dimensions and over time.
Looking at the big 3D picture.
Exactly. On a gene-by-gene scale, a crucial mechanism to focus enhancer-driven transcription and prevent transcriptional crosstalk is provided by special elements called insulators. These are specific DNA elements, which, in association with particular proteins, act as transcriptional boundary elements. Their function is to prevent an enhancer from acting on a promoter located on the other side of the insulator. Essentially, they delimit transcription domains, creating defined regions of gene regulation like molecular fences.
Fences to keep enhancers in their own yards.
A good way to think about it. We also know that precise DNA looping events, often involving these boundary elements, are vital for physically bringing distant enhancer and promoter sequences into close contact, enabling the precise communication required for regulation. The intricate rules governing these complex chromosome looping events are an area of intense research, but we know they're essential for establishing and maintaining order in the vast genome.
Okay, we've talked a lot about these regulatory proteins binding to DNA, whether they're activators or repressors. But how do they actually do it? What are the physical structures, the molecular hands or keys that allow them to recognize and interact with specific DNA sequences with such incredible affinity and precision? What's that precise molecular handshake look like?
This is a fundamental question, and it's where structural biology meets gene regulation. The incredible specificity and high affinity involved in transcription control are mediated by specific, recurring structural motifs within these regulatory proteins that physically interact with the DNA. Think of them as molecular tools, each specialized for binding. 3 key motifs account for many of these interactions: the helix-turn helix, HTH, the zinc finger, ZF, and the leucine zipper, LZ.
HTH, zinc finger, leucine zipper. Got it. What are the general rules for how these work?
Okay, before we dive into each, let's establish some general principles. First, these proteins must bind with very high affinity to their specific target DNA sites, but with very low affinity to all other nonspecific DNA. Otherwise, they just stick randomly everywhere and cause chaos. Second, it's fascinating that only small specific regions of the protein actually make a direct contact with the DNA. The rest of the protein is involved in other crucial functions, such as dimerization, forming multi-subunit complexes, creating contact surfaces for heterodimers, where different proteins come together, providing ligand binding sites, like for inducers or hormones, or interacting with coactivators, copressors, or the basal transcription machinery.
So only a small part touches the DNA.
Right. Third, the protein-DNA interactions are primarily maintained by non-covalent forces. Hydrogen bonds, ionic interactions, and van der Waals forces, the same fundamental forces that govern many molecular interactions throughout biology. Fourth, these motifs are quite class-specific, meaning if you identify one in a protein of unknown function, it strongly suggests that protein might bind to DNA. And finally, proteins with the HTH or LZ motifs typically form dimers, meaning two identical or similar protein units come together. Their respective DNA binding sites are often symmetric palindromes.
Binding is pairs.
Often, yes.
Huh?
Zinc finger proteins, on the other hand, often have repeated binding units. These dimeric or repeated structures allow for cooperative interactions between binding sites, significantly enhancing the degree and affinity of binding.
Okay, let's look at the first one. Helix turn helix.
Right, the helix turn helix HTH motif. This was the very first DNA binding motif whose three-dimensional structure was definitively discovered in the lambda CRO transcription regulator, which we just discussed as part of that crucial genetic switch. Each monomer of an HTH protein typically consists of 3 alpha helices. The crucial part for DNA recognition is the third alpha helix, often called the recognition helix. Its average diameter is perfectly sized, like a molecular key, to fit snugly into the major groove of B-form DNA, which is the most common form of DNA in our cells.
This fits right into the groove.
Exactly. The second and third alpha helices are held at an approximate 90-degree angle by a short turn of just 4 amino acids, forming the characteristic helix turn helix. When the protein forms a dimer, the recognition helices of each monomer project into successive half-turns of the major groove on the same surface of the DNA. Each monomer makes contact with about 5 base pairs of DNA. We see this dimeric helix-turn-helix structure not only in lambda CI and CRO, but also in CAP, the key MP receptor protein of E. coli, the tryptophan repressor, and many eukaryotic homeobox proteins, highlighting its conserved importance across different organisms and functions, a truly ancient and effective design.
Okay, next up, zinc fingers. What's the story there?
Okay, the zinc finger, ZF motif. This was the second DNA binding motif whose atomic structure was elucidated, and it's particularly prevalent in eukaryotes. It was initially characterized in a eukaryotic protein called TFI-IA, a positive regulator of 5S RNA gene transcription, which was known to absolutely require zinc for its activity. Structural analyses revealed that each TFI-II molecule contains 9 zinc ions. Each zinc ion is coordinated in a repeating complex, typically by a pair of closely spaced cysteine residues, followed 12 to 13 amino acids later by a pair of histidine residues. In other zinc finger proteins, such as the steroid-thyroid nuclear hormone receptor family, the histidine pair can be replaced by another cis pair. These fingers essentially lie on one face of the DNA helix, with successive fingers positioned in the major groove. Each finger typically contacts about 5 base pairs of DNA.
So the zinc acts like a tiny, indispensable scaffolding, literally holding the protein in the exact shape to grab onto the DNA.
Precisely. The zinc ion is absolutely indispensable. It provides the crucial structural integrity, stabilizing the protein's conformation so that it can correctly position the amino acid residues needed for sequence-specific DNA recognition within the major groove. Without that zinc, the finger collapses and the protein can't bind. And here's a truly surprising fact that shows its profound importance in human health. A single amino acid mutation in either of the 2 zinc fingers of the TWN25OH2D3 receptor protein, that's the protein involved in vitamin D metabolism, causes resistance to this vital hormone, leading to the severe clinical syndrome of rickets.
Wow. One tiny change, huge consequences.
That's a powerful and somber example of how a tiny molecular change affecting just 1 or 2 amino acids in this crucial DNA binding motif can have a devastating large-scale clinical impact on human health, highlighting the precision required.
And the third motif, leucine zippers.
Finally, we have the leucine zipper LZ motif. This motif was first identified in the mammalian enhancer binding protein CEBP. It's found in a 30 amino acid region at the carboxyl terminal end of the protein. This region forms an alpha helix where there's a distinctive periodic repeat of leucine residues at every 7th position. occurring for about 8 helical turns.
Leucines lining up like teeth on a zipper.
That's exactly the idea. This creates a distinctive zipper pattern of hydrophobic residues. What's remarkable is that this structure allows two identical or even non-identical monomers, like the proteins Jun and Phos, which often work together in cell growth to literally zip together into a coiled coil. This forms a very tight dimeric complex. This protein-protein interaction is crucial because it aligns and enhances the association of the separate DNA binding domains of each polypeptide with their specific target DNA sites. So, the leucine zipper itself is primarily a dimerization motif, bringing two DNA binding domains together so they can jointly and cooperatively interact with a symmetric DNA sequence.
So the zipper holds the protein partners together so they can bind DNA better.
Exactly. You find this motif in many eukaryotic transcription regulators, including GCN4, CEBP, FOS, Jun, and CREB, underscoring its widespread use in complex gene regulation.
Okay, this concept of modularity in these proteins, where they have distinct hands for different jobs, seems key. Does one specific part of the protein bind DNA, while a completely separate part is responsible for actually activating transcription? Or are these functions deeply intertwined? This seems like a fundamental question for truly understanding how these proteins work, especially if we want to master this.
This was indeed a critical, game-changing question, and it was definitively answered through a series of elegant domain swap experiments, pioneered in yeast with the GAL1 gene and its regulator, GAL4. The hypothesis was whether DNA binding and transcriptional activation were distinct functions performed by separable protein domains, or if they were inextricably linked together within a single functional unit.
Domain swap, like trading parts.
Exactly. Let's step with the experiment. The yeast GAL1 gene is involved in galactose metabolism, and its transcription is exquisitely controlled by the DNA-binding transactivator protein GAL4. GAL4 itself is a classic modular protein with a distinct N-terminal DNA-binding domain, DBD, and a separate C-terminal activation domain, AD. In the control experiment, the intact GAL4 protein binds to the GL1 upstream activating sequence, UAS, which acts as an enhancer in yeast and efficiently activates GAL1 transcription. As you'd expect, if you remove either the DBD or the AD or even the GAL1 UAS itself, that activation is completely abolished. All components are necessary for the normal function.
Makes sense. Need all the parts.
Right. Now, for the critical domain swap, researchers ingeniously took the DNA binding domain of GAL4, specifically the N-terminal 73 amino acids, and replaced it with the DBD of Lexa, an E. coli DNA binding protein that binds a completely different unrelated DNA sequence. This new custom-built protein, a chimeric protein, Lexa DBD fused to GAL4AD, initially did not activate GL1 transcription because the Lexa DBD couldn't recognize or bind to the GL1 UAS. No surprise there.
Okay, wrong key for the lock.
Precisely, but here's the truly crucial step. When the Lexa operator, Lexi's natural binding site, was inserted upstream of the GAL1 promoter, replacing the normal GL1 UAS, the hybrid Lexa-GAL4-AD fusion protein could now bind specifically to this new substituted Lexa operator. And remarkably, once bound, it activated GAL1 transcription.
Whoa. So it didn't matter which DNA binding part it had, as long as it could stick to the DNA near the gene, the activation part still worked.
Exactly.
So it's exactly like a Lego set. You can swap out the feet that attach to the DNA as long as the hands that do the activating are still there and they can properly reach the machinery. That's a brilliant and elegant way to demonstrate modularity.
Precisely. This experiment strongly demonstrated that the DNA binding domain and the transactivation domain are indeed distinct and can function completely independently. You can mix and match them. This modularity is a general characteristic of most transcription regulatory proteins, including repressor proteins, which also have separable DNA binding domains and silencing domains. The primary purpose of these molecules, whether they activate or repress, is to facilitate or block the assembly and/or activity of the basal transcription apparatus, that complex molecular machine, including RNA polymerase, on the linked promoter. It's all about precisely directing or preventing the access of the RNA polymerase machinery to the gene.
Okay, we've focused heavily on transcriptional control, that initial critical step from DNA to RNA. But you mentioned earlier that eukaryotes have many, many more regulatory points, a whole symphony of control. What else is happening to control gene expression after that initial RNA transcript is made? And how does that add even more fine-tuning?
Indeed. Eukaryotic cells employ a sophisticated array of mechanisms beyond just transcription, truly showcasing their evolutionary complexity. These additional layers of control offer even more fine-tuning and responsiveness that are generally not seen in simpler prokaryotes, often related to the extensive processing that eukaryotic RNAs undergo.
Like what kind of mechanisms?
Well, one of the most exciting and rapidly expanding areas of discovery in recent years is the profound role of non-coding RNAs, ncRNAs. These are ubiquitous RNA molecules that, as their name suggests, do not code for proteins, yet they contribute importantly to controlling gene expression. Among these, small ones like munes, microRNAs, and CERNAs, small interfering RNAs, which are typically only around 22 nucleotides long, are particularly well understood as powerful gene regulators.
Tiny RNAs controlling things.
Their mechanism of action is remarkably precise. They generally regulate specific messenger RNAs, mRNAs, by one of two main ways. If there are one or more mismatches in the base pairing between the miRNA and its target mRNA, the miRNA typically inhibits translation, meaning the mRNA is made but not efficiently turned into protein.
Slows down protein production from that message.
Right. However, if there is perfect base pairing between the miRNA and its target mRNA over all 22 nucleotides, it typically triggers mRNA degradation, essentially destroying the message. In a very few cases, miRNAs have even been shown to stimulate mRNA function, adding another layer of complexity to their roles.
So these tiny RNA molecules are like precision missiles, targeting specific messages, and either silencing them or destroying them. It adds a whole new, incredibly subtle dimension to gene control, beyond just turning genes on or off at the DNA level.
It absolutely does. The action of a single miRNA can lead to dramatic and widespread changes in protein production and, consequently, in overall gene expression. From a biomedical perspective, these small ncRNAs have been implicated in numerous human diseases, including heart disease, various cancers, muscle wasting disorders, viral infections, and diabetes, making them significant and promising targets for therapeutic development.
Huge potential there.
Definitely. Like DNA-binding transcription factors, miRNAs and CERNAs are transactive molecules. Once they are synthesized and properly processed, they interact with specific protein complexes and then bind to their target mRNAs based on normal base pairing rules, dictating the mRNA's fate. The full magnitude and universality of ncRNA-mediated gene regulation are still being actively investigated, but their importance in the intricate network of gene control is undeniable and growing.
Okay, what else besides transcription in these small RNAs? You mentioned more dramatic things earlier.
Yes. Sometimes, to get enough of a certain gene product, the cell doesn't just turn up the volume of its expression, it actually makes more copies of the gene itself, or even physically rearranges its DNA. These are indeed fascinating and dramatic forms of regulation that involve fundamental changes to the genomic DNA itself, showcasing the cell's remarkable genomic plasticity.
Gene amplification. Making more copies.
Right. Gene amplification refers to increasing the number of gene copies to meet very high cellular demands for specific molecules. For example, ribosomal RNA encoding genes are often present in hundreds of copies in the germline of many organisms, meaning this high copy number is transmitted across generations, ensuring a plentiful supply of ribosomal RNA for protein synthesis, something constantly needed. A classic example during development is in Drosophila, fruit flies, oochogenesis, where genes for chorion, eggshell proteins, are selectively amplified through a process of repeated initiations during DNA synthesis, but without full chromosome replication. This rapidly provides multiple transcription sites, allowing for massive rapid production of eggshell proteins needed for egg development.
A specialized need met by making more gene copies.
Exactly. However, there's also a dark side to gene amplification that's highly relevant to human health. In cancer cells, drug resistance can devastatingly arise from the amplification and increased expression of genes encoding proteins that either degrade therapeutic drugs or actively pump them out of the cell. This is a significant challenge in cancer treatment, as the cancer cells effectively evolve drug resistance by amplifying the genes that help them survive the therapy, making once effective treatments useless.
A major problem in cancer therapy. And gene rearrangement.
Second, gene rearrangement involves physical changes to the DNA sequence itself, and it's particularly important for generating diversity, especially in our immune system. The most spectacular example is with immunoglobulin genes, which encode antibodies, the molecular warriors of our immune system. Antibodies are made of heavy and light polypeptide chains. Their coding sequences are not contiguous in the germline DNA. Instead, they are encoded by several different modular segments, variable V joining J and constant C segments. For instance, in the IgG light chain, there are roughly 300 VL segments, 5 JL segments, and 10 CL segments, all tandemly repeated in the same chromosomal region like a massive molecular library.
So the blueprint for one antibody isn't a single continuous stretch of DNA, but a collection of modular parts, like a molecular Lego set for immunity that needs to be assembled. That's wild.
Exactly. To produce a functional IgG light chain, a single VL, JL, and CL coding sequence must be recombined to generate a single contiguous transcription unit. This involves a precise process of selective DNA recombination that literally deletes all the unused segments. For example, if there are 300 VL segments, only one is chosen and physically stitched together with a JL and CL segment, with the other 299 VL segments and other unused JL and CL segments being excised from the chromosome and discarded.
Cutting and pasting the DNA itself.
Precisely. Furthermore, after this recombination, the VL sequences undergo additional point mutagenesis targeted single nucleotide changes to generate even more variability, hence the name variable region. This incredible directed genetic rearrangement occurring within a single clonal population of B cells provides the immune system with an almost unimaginable repertoire of sequences, literally millions of combinations. It enables both the extraordinary immunologic flexibility required to respond to a vast array of diverse pathogens we might encounter, and the exquisite specificity needed to target particular antigens. Each B cell clone can thus produce a unique antibody tailored to a specific threat.
An amazing system for generating diversity.
It's truly a masterclass in genomic plasticity for a functional purpose, allowing for an adaptive immune response.
Okay, so once we have that initial RNA transcript, the story still isn't over. What else can happen to it before it becomes a protein? I understand there are different ways to process it, almost like editing the same raw blueprint in different ways to build distinct versions of the same product.
Yes, that's absolutely right, and it's a powerful control mechanism largely unique to eukaryotes. Alternative RNA processing allows a single gene to produce multiple distinct protein products from that same initial RNA transcript. This vastly increases the functional output of our relatively limited number of genes. This amazing versatility can result from the alternative use of three key elements.
Where are they?
First, alternative transcription start sites, promoters. A set gene can actually have multiple promoters located at different points along the DNA. Depending on which promoter is activated and used, the resulting messenger RNA will have a different 5' exon at the very beginning of its coding sequence. This can lead to different protein isoforms with different N-termini, which in turn can impact their function, their stability, or even their localization within the cell. Classic examples include mouse amylase and myosin light chain, and rat glucinase, all demonstrating different 5' ends leading to functionally distinct protein variants.
Starting the message in different places.
Right. Second, and perhaps the most widely recognized form, is alternative intron exon splice sites. After the initial RNA transcript is made, it contains both exons, the coding regions, and introns, non-coding regions that must be removed. Through alternative splicing, different combinations of these exons can be included or excluded from the final mature mRNA. This means that from one primary transcript, the cell can cut and paste the exons in different ways to produce multiple distinct mRNAs, each encoding a different protein isoform.
Mixing and matching the coding pieces.
Exactly. A classic example is the atropomyosin gene, which astonishingly produces 7 unique atropomyosin mRNAs in 7 different tissues, all from a single gene. This highlights precise tissue-specific regulation at the splicing level, allowing a muscle cell to make one form while a brain cell makes another.
One gene, many proteins via splicing.
And third, we have alternative polyadenylation sites. The poly-A tail is a string of adenosine nucleotides added to the 3' end of most eukaryotic mRNAs, which is vital for mRNA stability and efficient translation. However, a single gene's primary transcript can sometimes use different polyadenylation sites. This leads to mRNAs with different 3' ends, which can profoundly impact mRNA stability, its localization within the cell, or even change the C-terminus of the encoded protein if the alternative site is upstream of a coding sequence.
Changing the tail end of the message.
Right. For instance, the O-immunoglobulin heavy chain primary transcript can use 2 different polyadenylation sites, producing mRNAs of 2,700 bases or 2,400 bases. The critical functional consequence of this subtle length difference is that the MR protein remains attached to the B lymphocyte membrane, where it acts as a B cell receptor, while the shorter MR immunoglobulin is secreted as a circulating antibody. This single alternative processing event determines whether the antibody acts as a cell surface receptor or a free-floating immune molecule in your bloodstream.
So it's like taking the same raw architectural blueprint and then, through different editing decisions, cutting and pasting it in different ways to build distinct versions of the same product, depending on what the cell specifically needs, That's incredibly efficient, maximizing information from a single gene.
It absolutely is, and it significantly expands the functional proteome. The total set of proteins an organism can produce far beyond the number of genes in the genome. The precise mechanisms and regulation of these complex splicing and processing decisions are still areas of very active and exciting research, representing a frontier in understanding how life creates such diversity from shared genetic information.
And even after mRNA is processed and ready to be translated, its fate isn't sealed, is it? It can be quickly destroyed or hang around for a long time, further fine-tuning how much protein is actually made, so the cell can control the lifespan of the message.
That's entirely correct. The regulation of messenger RNA stability, or its half-life, is yet another crucial regulatory point, often overlooked but profoundly impactful. While many mammalian mRNAs are quite stable, with half-lives measured in hours, some turn over very rapidly, lasting only 10-30 minutes, this directly impacts the amount of protein that can be produced from that mRNA molecule. If an mRNA has a short half-life, less protein will be made from it over time, even if transcription is high. If it's stabilized, more protein can be made from it.
Like a self-destruct timer on the message?
Kind of, yes. The mechanism largely involves the fact that mRNAs exist in the cytoplasm not as naked strands, but as complex ribonucleoprotein particles, RNPs. Here, various proteins can either protect the mRNA from degradation by nucleases, enzymes that break down RNA, or conversely promote nucleus attack. It's thought that the interaction of these RNP proteins with specific structures or sequences within the mRNA dictates its stability or destabilization.
Proteins protecting or targeting the mRNA.
Exactly. The ends of mRNA molecules are particularly important for stability. The 5-cap structure in eukaryotic mRNA acts like a protective helmet, preventing attack by 5' exonucleases, which degrade RNA from that end. Similarly, the poly-A tail, typically 100-200 nucleotides long at the 3' end, minimizes the action of 3' exonucleases. For mRNAs with these protective structures, it's generally thought that a single internal endonucleolytic cut a precise break in the middle of the mRNA can then allow exonucleases to rapidly digest the entire molecule. Other structures or specific sequences within the 5' untranslated region, 5' UTR, the coding region itself, and particularly the 3' UTR, can also either promote or prevent this initial endonucleolytic action. And critically, various cellular effectors like hormones or stress signals can regulate mRNA stability by altering the amount or activity of these mRNA binding proteins.
So the cell can fine-tune protein production not just by how much mRNA it makes, but also by how long that mRNA sticks around and is available to be read. It's truly a multi-layered masterpiece of control, optimizing every step.
It truly is. This coordinate regulation of mRNA synthesis, processing, localization, and stability, along with those more fundamental changes like gene amplification and rearrangements, confers remarkable adaptability on the cell. It's this integration of control mechanisms at virtually every step of gene expression, from the initial DNA blueprint to the final protein product, that allows for such incredibly precise and dynamic responses to internal cues, external environments, and the complex demands of development and daily life.
We've covered so much ground today, from the elegant simplicity of bacterial operons, those incredible molecular energy managers, to the mind-boggling architectural feats of enhanceosomes in our own cells, literally bending DNA to create perfect docking stations. And we navigated the intricate world of RNA processing and mRNA stability, controlling the very lifespan of a message. It's abundantly clear that gene expression is not a single switch, but an entire orchestra playing a complex dynamic symphony.
This deep dive has shown us that virtually every normal somatic cell has the same genetic blueprint, but their unique functions and their remarkable ability to adapt are dictated by the dynamic differences in which genes are expressed, where, and when. We've seen how cells respond to environmental changes, developmental cues, and physiologic signals by modulating gene expression at multiple integrated levels, from the initial transcription through mRNA processing, localization, and stability, as well as more fundamental dramatic changes like gene amplification and rearrangements of the genome itself.
And we've truly seen how specific protein-DNA interactions, often involving elegant modular domains and fascinating structural motifs like the helix-turn helix, zinc fingers, and leucine zippers, drive these controls with incredible precision. Plus, we unpack the pivotal role of chromatin modifications, what we call the histone code, and DNA methylation in eukaryotic regulation, creating that crucial dynamic accessibility to our genes. And let's not forget those remarkable non-coding RNAs, silently orchestrating translation and mRNA stability, adding yet another layer of fascinating subtle control.
This extraordinary molecular precision and the sheer number of control points, each influencing a gene's fate, truly highlight the immense power and subtlety of life at the molecular level. It raises a profound question for you, the listener, to consider. Given this extraordinary molecular precision and the numerous points of control, what are the ethical and practical implications of developing technologies that could precisely alter any of these levels of gene expression? How might that fundamentally change our approach to medicine, development, or even human identity itself? What new avenues of scientific understanding and technological innovation might open up as we continue to unlock the deepest secrets of these multi-layered gene expression control systems? Think about the possibilities that arise from truly mastering this fundamental knowledge.
A powerful thought to end on. We hope this deep dive has given you a newfound appreciation for the incredible complexity and elegance of life at the molecular level, and arms you with knowledge to truly feel like an elite professional in this fascinating field. Join us next time for another deep dive into a world of surprising facts and profound insights.